Hi class, in this video we're talking about biopsychology. There's going to be two main things we go over. We're going to talk about genes first, and then we'll spend the majority of this chapter talking about the nervous system, partly the peripheral nervous system, most of the time the central nervous system. But first, genes. When we talk about genes, a big part of this is natural selection. The theory of natural selection says that organisms that are better suited for their environment will survive and be able to reproduce, while organisms that are not suited for their environment will die off before they're able to reproduce. And so when we see a specific gene in the environment, in organisms, lots of organisms, you have to assume that there has to be some benefit to that gene, because if it was only detrimental and not beneficial, it would not survive in that gene pool. Take, for example, sickle cell anemia, which is a genetic condition where it's a recessive trait. And we'll get into the whole dominant recessive thing, but you need a sickle cell gene from both parents to have sickle cell yourself. If only one parent gives you a sickle cell gene, you are called a carrier, but you do not have full-blown sickle cell. And sickle cell anemia, if you have this, it means that your red blood cells are shaped like crescents, like sickles, and it can cause a whole host of problems. It clogs in your blood vessels. It can, block blood flow, causes a lot of pain. It can be fatal. And so why do we still see this disorder? Why has this gene stuck in the gene pool if it causes sickle cell anemia? According to natural selection, there has to be some benefit to this. And what they have found is that people who are carriers of sickle cell, who have one of the genes but don't have full-blown sickle cell, are immune to malaria. So sickle cell anemia is a disease you usually see in people of African descent. Malaria kills millions of people a year, more than sickle cell kills. And so because people who are carriers are not going to die from malaria, that is how that gene has stayed in the gene pool. So whenever you see a gene that continues in the gene pool, there has to be some benefit to it, according to natural selection. When we talk about all these differences in genes, that is genetic variation. Genetic variation is the genetic difference between individuals. So it's what contributes to our ability to adapt to our environment. You should know the difference between chromosome, DNA, and genes. Chromosomes are your long string of DNA. DNA are those helix-shaped molecules made up of your nucleotide base pairs. And then genes, which are in a chromosome, are made up of sequences of DNA. So it goes chromosome, genes, DNA, for biggest to smallest. Your genes are what control or partially control a lot of your visible characteristics. We call these our traits. And so when you say this gene connects to this trait, you're talking about the allele. So alleles are specific versions of a gene that will then lead to a trait. When you think about this, you need to know the difference between a genotype and a phenotype. Your genotype is your genetic makeup. It's what your actual genes are. Whereas your phenotype is your inherited physical characteristics. You can always tell someone's phenotype. You can see someone's phenotype. You can't always see someone's genotype. And for most traits are going to be what we call polygenic. And so what that means is that there's lots of genes that control one trait. It's not just one gene. There are some traits that are controlled by only one gene, but a lot of times it's multiple genes. And again, when we talk about these genes, you need to know the difference between recessive and dominant. And so a recessive gene is one where you need two versions of the recessive gene in order for that trait to be visible in your phenotype. If it's a dominant allele, that means that you only need one copy for that genotype to be visible in the phenotype. So when you look at someone's phenotype, you can only know what their genotype is if they have a recessive phenotype, because that means you know they have all of their alleles connected with that trait are recessive. If they have a dominant phenotype, you don't know if their genotype is homozygous, where they have only dominant alleles, or if it's heterozygous, where they have dominant and recessive. So a few examples of traits that are where it's only one allele, where it's not polygenic, it's only one, so that you can get this idea between the phenotype, genotype. If you put your hands together like this, which thumb is on top? If your right thumb is the one on top of your left thumb, that is a recessive trait. So for example, that's the way I am. My right thumb goes above my left thumb when I put my hands together. The other way, it feels weird. I can't do it. So because my phenotype is right over left, you know my genotype. You know I have two right over left thumb genes. So because I have this recessive phenotype, you know what my genotype is. For all of you that go the other way, that your left thumb goes on top, you don't know what your genotype is. You don't know if you have two left thumb on top genes or one left thumb, one right thumb gene. Another example, if you can roll your tongue, and this is one I cannot do. If you can roll your tongue, that is a recessive trait. It means you only have two recessive genes. If you can't roll your tongue, then you don't know if you are heterozygous or homozygous for no tongue rolling. And then another one that also shows an extra difference is if you're colorblind. So if you're colorblind, any form of being colorblind, that is a recessive trait. However, that gene for colorblindness is a sex-linked gene, what we call a sex-linked gene, which means that you have it on your X chromosome. There's probably a couple of Y chromosome sex-linked genes, Y being what makes someone genetically male. Everyone has an X. If you have two X, you're genetically female. So colorblindness, the colorblind gene is on the X chromosome. It's a recessive trait. If you are a man, though, if you're genetically male, you only have one X chromosome, which means if you get that one X, for colorblindness, you will be colorblind. You can't be heterozygous. You only have one of the genes. For people who are genetically female, because you have two X chromosomes, if you get one recessive colorblind gene, you still have another X chromosome that can have the dominant no colorblind gene. And so you see significantly different rates of colorblindness for people who are genetically male versus people who are genetically female. You are significantly more likely to be colorblind if you are genetically male, because you don't have the option of another dominant gene coming in to stop it. So it's much more rare to see people who are genetically female who are colorblind. They exist, but it's less common. So one way that we get, final definition here, one way that we get these changes in our environment is through mutations. So mutations are sudden permanent changes in the gene. If it's a harmful mutation, then it can potentially be fatal. It's not going to get passed on. If it is a beneficial one, then you're going to see it get passed on to future generations. And so a lot of the changes we see in our gene pool are because of mutations that happened that ended up being beneficial for people. When we talk about genes and we talk about it in terms of psychology, a big thing we tend to think about is the gene-environment interaction. Go back and think about from chapter 1 that biopsychosocial model. If you're going to understand something, think about biologically, psychologically, environmentally, socially, what's going on. So that includes this research as well. We like to think about range of reaction as well as epigenetics. Your range of reaction is the idea that our genes set sort of a boundary, a spectrum within which you can operate, and then it's your environment that interacts with genes to determine where on that range you will fall. One example of this would be height. Your genes give you a possible range for what your height can be. For example, I'm not even 5'3", my husband's 5'6". There is no chance our children are going to end up being like 6'5". They did not get the genes to be 6'5". It's never going to happen. However, the genes we gave them could be a range where they could end up anywhere between 5'4" and 5'8". Where on that spectrum they're going to fall is going to depend on their environment. So, do they get lots of nutritious food? Do they get lots of exercise? Do they have a healthy environment? Then they'll reach the upper limit. They'll be closer to 5'8". If they are malnourished, then they'll be closer to the 5'4" range. And so that's why if you go back a few generations, people tended to be shorter. They didn't have as good of nutrition, and so they weren't going to reach the upper limit of their potential. So that's range of reaction. Here is your potential range. Your environment's going to decide where on that range you fall. So your genes set the limit, environment determines where you fall. So the idea of epigenetics is a little bit different. Epigenetics looks beyond the genotype and studies how the same exact genotype can be expressed in different ways. So you have the same genotype, but different phenotypes. This is different than range of reaction because the genotype isn't as fixed or limited in the epigenetics idea. It's basically your environment turning genes on and off. You could think of it that way. So a good example of this is with schizophrenia. And so this table here is connected with people who were diagnosed with schizophrenia, potentially. In this study, what they did was they looked at kids who were adopted, and they looked at whether their biological parents had schizophrenia. That would be people with high genetic risk, because we know it's partly a genetic disorder. So low genetic risk, it means their biological parents were not diagnosed with schizophrenia. And then the environment they were raised in their adoptive homes. So a disturbed environment or a healthy environment. If you had that high genetic risk and a disturbed environment, over a third of those children developed schizophrenia themselves. However, if they had that genetic risk but they had a healthy environment, less than 6% developed schizophrenia. So that is that epigenetics. That disturbed environment basically turned on the gene for schizophrenia. All these kids in the high genetic risk group had the same genotype. They all had that genetic basis for schizophrenia within them, but it was their environment that determined whether it got turned on and off. And for those with a low genetic risk, 5.3% if it was a not good environment, 4.8% for healthy environment, which all of these are within a point of each other. It's that combo that sort of flipped the switch and turned it on. The next thing we're going to talk about is the nervous system. So we're done with genes, we're moving on to the nervous system. First definition is, what is your nervous system? So I can make sure I add that in. Your nervous system is composed of two basic cells and is responsible for communication within your body. The two cells that make up the nervous system, you have glial cells and neurons. So first, glial cells. Glial cells are the scaffolding for the nervous system. They're sort of the worker bees of the nervous system. They provide insulation for the neurons, they transport nutrients, they take waste away, they help with the immune response, lots of things. The original idea was that there were 10 of them for every one neuron. Researcher Susan Herculin found that it's closer to 1 to 1, that there aren't really that many more glial cells. It's more of a 1-to-1 ratio for glial cells and neurons. Next, what is a neuron? Neurons are the communication cell in the body. They are interconnected information processors that are essential for all the tasks in the nervous system. They are what send information through the nervous system. You have about 100 billion neurons in your body when you are born, and you need to know the parts of the neuron. So we have a neuron here, and I am going to go through these parts. For a neuron, you have, and so for this, by the way, as I talk about this, information always only moves in one direction for a neuron. So information is moving that direction. That's the way it's always going to work. So it always comes in on over here, comes in through the dendrite, and leaves out through the terminal buttons, always. And later we'll talk about the different types of neurons. No matter what type it is, whether it's a motor neuron, sensory neuron, or an interneuron, they all have the same components. They might look a little different, but they all have the same components. So, the parts of a neuron. Again, information enters a neuron through the dendrite. So dendrites receive information from other neurons. Then that information will go through the soma or cell body. The cell body, or soma, is the control center for the neuron. That will decide, is this a message we're going to carry on? If it's a message that's going to get passed on, it's going to go down the axon. The axon is this long tube-like structure. It is the longest part of the neuron. We'll talk later about other stuff that happens in the axon. The axon is covered in myelin sheath. The myelin sheath covers the axon and helps with insulation. So the myelin sheath is a fatty substance that insulates the axon. And then finally, after it leaves that axon, the end of the axon has terminal buttons. So terminal buttons are what send the message out to the other neurons in line. So dendrite, soma, axon, cell body. That is always going to be the order it goes in. Okay, so let's keep going. For a neuron, there are two ways that the neuron communicates. You have between neuron communication, so how one neuron sends a signal to the next neuron in the line. And then you have within neuron communication, how a signal goes from the dendrite to the terminal button. So first we're going to draw, I'm going to explain the within neuron communication. So what happens in that axon, basically. And for this, that is an electrical process. So you have an electrical signal within the neuron, and then it's a chemical signal between the neurons. So what I'm going to do is draw a picture of an axon. So there's my axon. I'm not going to draw the myelin sheath, so it's easy. And so what you have here, and I'll go through these definitions as well. You have first the membrane potential. This is the difference in charge across that membrane in the axon. So you have a different charge within the axon versus outside of the axon. And this is because of the ions, those charged molecules that are either in the fluid in the axon or the fluid outside of the axon. So when a neuron is in its resting potential, what that means if it's in its resting potential is it's not working. It's resting. It's not doing anything. It's not sending a signal. If it is in its resting potential, it has a negative charge. And so what you have is you have a lot of, when it's in its resting potential, you have a lot of sodium, which is positively charged ions, outside of your axon. Big old fat sodium ions. And you have a couple of potassium, small, and I'll do that as a different color so that it's less confusing. You have a few potassium ions inside, but not as many. More of the big fat sodium outside. And so you end up having more negative outside, or more positive outside, so it's a positive charge outside compared to inside. This is the resting potential. And then what happens is that you have the signal that comes through the dendrite, it goes to the cell body, that soma, and the neuron says, yes, this is something we're going to pass along. And so it has to reach its action potential. The action potential is when you have that enough of a positive charge within the neuron, it switches its charge, so that the electrical signal will go through the axon and go to the next cell in line. To reach that action potential, you have to hit the threshold of excitation. The threshold of excitation is a certain level that a neuron has to reach, that certain positive level, so that it becomes active and it can reach that action potential. The action potential can start. So you have all of these negative ions that'll flood into the axon. Or not negative. You have all these positive ions that are going to flood into the axon so that it can start. So what happens is, basically, do this a little differently. You have a bunch of holes that open in the axon. And it's going to, these pumps that let the sodium into the neuron. So once all the sodium is here, it switches the charge from negative to positive, because now all of these sodium ones have moved in. And once this hits a positive enough charge, that's when that threshold of excitation has been reached. That's when the action potential is going to start. It's going to send the message to the next one in line. After that happens, the message has been passed on. This needs to reset. The way that happens is that you get the potassium gets pumped out so that this can reset to a negative charge and you can be done, go back to the resting potential. So in terms of, I'm actually going to draw a little thing here. And I'm going to have, that is 0. That is neutral. So we'll have that red line be neutral. So when it's in, well, I'll do this a different color so it's easy to tell. When this is in its resting potential, it has a negative charge. Then that message comes in through the dendrites. Yes, this is something we're going to get sent on. All that sodium gets pumped in. It hits that threshold of excitation. The signal gets passed on. Then it has to reset. All that sodium is going to get pumped out. It actually gets hyperpolarized for a second and then resets to normal. And now it's back at the resting potential and it's ready so that it can go and do the next one in line. So that is the within neuron communication. And final thing here, that all or none. When this happens, it is literally all or none. If you don't get enough sodium pumped in so that it only gets to like that level and it doesn't hit that threshold of excitation up there, it doesn't send the signal. It just resets and goes back to normal, but no signal gets passed on. It stops here at this neuron. So it's an all or none thing. It sends the full signal or it sends nothing. It can't send half of a signal. So again, that is the within neuron communication. The final thing I'll talk about in this video is the between neuron communication, and then we'll be done with this video. So for the between neuron communication, I don't have the words here, so I am going to put them here now. So these are all the words I'm about to go over. And I am going to now, I'm going to draw a neuron and another neuron. I'm going to draw two neurons talking to each other, and we're going to go through all of the things that are connected with this. So this is the terminal button. And on the end of the terminal button, this orange thing is what we call a synaptic sac. And inside the synaptic sac are neurotransmitters. Neurotransmitters are chemicals that are used with between neuron communication. So they are chemical messengers. So terminal button, synaptic sac or synaptic vesicle, neurotransmitters. That's all on the terminal button side because the information is moving in this direction. So it goes out the terminal button into dendrite. So over here we have the dendrite. And on the end of the dendrite, you have receptor sites. Receptor sites at the end of the dendrite. Actually, maybe that a different color. And one more definition before we get into exactly what's happening here when a message comes through. You will notice right here, these two neurons don't touch. Two neurons never directly touch each other. There's always a gap between neurons. This is called the synapse or synaptic gap. It is the fluid-filled space between neurons. So what happens when the message comes through that axon down through the terminal button, and this is a message that's going to get passed on? What happens is that the synaptic vesicle opens, synaptic sac, synaptic vesicle, and these neurotransmitters go across the synapse and into these receptor sites. They fit like a puzzle piece. And that is what signals the message to now go through this neuron. After that happens, this whole process has to reset itself. The way it resets itself is reuptake Well, one of two ways. Either these neurotransmitters actually just get absorbed into this neuron, or they leave these receptor sites and they go back home again. They return back to the terminal button, back into their synaptic vesicle, and it seals itself back up again. And now it's reset, it's ready to go. So if the neurotransmitters return back to their synaptic vesicle, that's what we call reuptake. And now this is all ready, it can start the whole process over again. So again, you have the end of the terminal button, synaptic sac filled with neurotransmitters. You have the synapse in between the two neurons. You have receptor sites on the dendrite side. And when those neurotransmitters return back to their synaptic sac, that is called reuptake. So that is between neuron communication. And that is where we will end this video. In the next one, we will start with the types of the nervous system. We'll start with the peripheral nervous system.